Good morning, everybody, and thank you for joining today's webinar on our next generation tools to clone, express, and purify proteins. I'm Malti Rahman. I'm a product manager for cloning and protein from Takara Bio Europe, and I'll be presenting today's webinar. So if you have any questions during the webinar, please just type them into the chat box and we can take all the questions at the end. So this webinar will also be recorded, so we will send you a link to the recording also after the webinar. Okay, so before I talk about the technologies themselves, I first wanted to present you with the typical clone, express, and purify workflow that most researchers use for their protein expression and purification experiments. The first step, of course, is to clone your target gene that you're interested in studying into your protein expression vector of choice. So most people are doing this via traditional ligation-based cloning methods, which can sometimes be really time-consuming and laborious. And today I'd like to present you with an alternative system, our next-generation infusion system, which we believe is just a faster and better way to clone. So once you've generated your protein expression vector, the next step is to express the target protein. And usually the target protein is expressed with a tag such as the 6-his tag, which is used by most researchers because it's small and it's well tolerated. Takara do offer a wide range of different protein expression systems, and I will briefly present them today, but this won't be the main focus of my talk. But if you're interested in finding out more, you can always visit our website. So once the target protein's been expressed with a tag such as the his tag, the next step is to purify the target protein before you can then use it in your downstream functional studies. So most researchers are using resin-based methods to purify his tag proteins, but today I'd like to present you with our next generation Capture-M technology, which is a membrane-based purification technology as a better and faster alternative to resin-based methods. So, as I said before, the first step is to choose the expression system or vector that you want to use, okay, and then to, of course, clone your target gene into the relevant vector of the relevant expression system. So from Takara, we offer many different systems, including bacterial systems, so this includes E. coli systems, bacillus systems. We have insect expression systems, so our baculoviral system. We also have some nice mammalian systems, including our PHEK293 ultra vectors. And finally, we also have our human cell-free system, which is an in vitro transcription and translation system. And as you can see, as you go from bacteria to the human cell-free system, the number of post-translational modifications that are possible also increase. So how do you best choose which expression system is the best one for your experiment? Well, this table gives you a nice overview of the different systems that we offer and the main features, considerations to take into account when choosing your system. So for example, if you want a simple low-cost system which has a high yield, then we would recommend our E. coli PET Express and Purify system, which will allow you to obtain up to 1 gram per liter of protein, and it's also very cheap. So this is really the first choice system, and it's an IPTG inducible system. However, if your protein is more temperature sensitive, then you could consider using our P-cold vector system, which will allow you to express the protein at 15 degrees C. But of course, the yield is not as much as the PET Express system. We also have a nice single protein production system. So this allows you to just express your protein within bacterial cells. So this is a really elegant system. I won't talk about this today, but if you're looking for a very pure protein, this system could be good. Although again, the yield is not so high. Then we have our more specialized Brevibacillus and B-Cetillus systems. So these are good if you're really looking for high yield protein, but also if you're looking for a system that will allow you to express secreted protein. So in fact, with Brevibacillus, this is optional. You can use the vector with the secretion tag or without, it's up to you. And with the B-Cetillus system, you can actually optimize secretion. And these systems are also good because you get active protein, and it allows certain bond formations, such as disulfide bond formations. Okay, and in fact, B. subtilis is the best one for large-scale protein production experiments. We also offer our insect baculoviral system. So this is good if you want more post-translational modifications, because of course this is closer to mammalian cells, and you want a good yield, so you can get up to 0.5 gram per liter. And it's also good for temperature-sensitive proteins. But moving more towards the mammalian system, we have our PHEK293 ultravectors, which allow you to express your target protein in 293 cells. So you can get up to 10 mg per liter, which is pretty good for mammalian cells. And also the protein is secreted, and of course you will obtain most post-translational modifications with these vectors, since you're expressing in 293 cells. And finally, we also have our human cell-free in vitro transcription and translation system, which is particularly good if you're trying to express toxic proteins. So if you're just not able to express it in bacterial or insect or mammalian cells, you could consider this system. And with our system, you get a better than average yield of 50 milligrams here. And it's also good for temperature-sensitive proteins. Okay, so once you've decided the expression system that you want to use, i.e. the vector that you want to clone into, the next step is to actually clone your target gene into this expression vector. So now I want to present you with our next generation infusion system as a better alternative to traditional ligation-based cloning. So you're all aware that cloning usually represents the first step of most molecular biology experiments carried out in labs today. But it's not usually the main focus of the experiment, rather just a simple step on the way to you actually running your more interesting functional experiments. So what if you could have a fast and accurate method which would allow you to minimize the amount of time spent on this step? Well, we believe we have such a system in our infusion system because it can be used to easily clone single inserts, multiple inserts in a single reaction, to easily do site-directed mutagenesis, and also to easily add C or N terminal tags. We believe it's really a cloning system with no limitations that will allow you to get the right clone every time. Okay, so you might be asking yourself, so what's so great about this infusion cloning system? Well, the beauty of infusion lies in the fact that it's completely sequence independent, so it can be used to clone any insert into any vector at any locus. You're not restricted either by the sequence of your insert or by the sequence of your vector. It can also be used to directly clone into your final destination vector, so you don't need to do any sub-cloning steps, which can sometimes be the case, for example, if you're using TA cloning methods where you just capture the PCR product, but then you still need to do a downstream sub-cloning step. It's also seamless and directional, so this means that no extra bases are added to your sequence unless you want to add them. So this is in contrast, for example, to gateway recombination cloning, where the recombination sites often add additional unwanted amino acids to your protein of interest, which can sometimes alter the function of the protein. So this is not the case with the infusion system. You can also do some really nice applications such as cloning multiple fragments in a single reaction, as well as doing mutagenesis in a single reaction. So you don't need a separate kit for mutagenesis if you use infusion. Okay, so how does the infusion system work exactly? The first step is to decide the vector that you want to clone into, which of course can be any expression vector that you have in your lab. You simply you linearize it at your desired locus that you want to clone the insert into. So this can be either via restriction digestion if you have suitable enzymes, or by inverse PCR if you want to be completely restriction site independent. And as long as you use a high fidelity polymerase for the linearization for the inverse PCR, this should be fine, such as our CloneAmp Hi-Fi premix, which is included as part of all our Infusion Plus systems. You just simply need to know the 15 base pairs found at either end of your linearized vector, so shown in red and yellow here, because these same 15 base pair overlaps will be added to either end of your insert, which can also be any insert. Okay, so you will you would generate your insert via PCR using gene-specific primers that also carry these 15 base pair overlaps to either end of your vector. The insert and vector are then incubated with the infusion enzyme for 15 minutes at 50 degrees C. And what the infusion enzyme does is it will fuse the insert to the vector at these 15 base pair overlapping regions. This, upon transformation into E. coli, will result in gap repair and positive clones containing the recombinant vector. Now, the beauty of infusion is that you're not limited by the sequence of your insert because you don't digest your insert at all. Because normally with traditional ligation-based cloning, you need to make sure that the enzymes that you use to digest your vector do not cut within your insert. Well, this is not an issue with infusion. So this really gives you a lot of freedom. And also you do not need to dephosphorylate your vector at all. So the number of steps involved in the cloning protocol are also are fewer, so it's more streamlined compared to traditional ligation-based cloning. Now, one important point to mention is that primer design, of course, is critical for infusion cloning because you need to add these overlaps to your insert, which are homologous to the linearized vector. And so I would strongly recommend you to use our new online infusion cloning primer design tool to help you with this important step. Now, let me go into more detail of exactly how the infusion enzyme works. Okay, so if you imagine that this is the insert, okay, with the 15 base pair overlaps, which are homologous to the ends of the linearized vector, shown in red and blue here. So if you imagine this black region is just missing. And what happens when you incubate the infusion enzyme with the insert and vector is that the 3' exonuclease activity of the enzyme chews back the 3' ends of the vector and the insert, allowing the insert to spontaneously anneal to the vector at these exposed 15 base pair overlapping regions. Now, at this stage, the insert and vector are held together just via hydrogen bonds. It's only when you transform this into E. coli that you get gap repair occurring in vivo in the E. coli cells, and you get permanent covalent linking of the insert to the vector happening within the E. coli cell, and thus you get positive clones containing your recombinant vector. What are the advantages of using infusion over other methods? The most important advantage, of course, is cloning success, and this is because we've spent over 10 years of progressive research into perfecting this enzyme, which allows you to clone directly into any vector, and the cloning is also seamless. Infusion has an excellent performance. We really obtain over 95% positive recombinant clones or cloning efficiency for a wide range of insert sizes. So anything from 20 base pair oligos to 15 kb inserts. You simply need to screen 2 clones per construct, and both should be positive. This is in contrast to, for example, ligation-based cloning, where on average most researchers screen at least 10 to 20 colonies per construct. So you can really save time and money because of the high efficiency of infusion cloning. It's fast and accurate. It's also simple and powerful, so you can use it to clone large or long inserts as well as single and multiple inserts. So the graph on the right here shows where we used infusion to clone 8 kb, 12 kb, and 15 kb inserts. So you can see, although the total number of colonies does drop as you clone longer inserts, which is normal because you're putting more pressure on the cell, the percentage of positive clones doesn't change much. It remains high at 90 to 100%. And it's really one system with multiple applications. This slide just shows you the accurate, directional, and seamless nature of infusion clones. So this shows sequencing data of some positive clones that we generated using infusion. So firstly, you can see that all the sequences are error-free, which shows that the included clone amp-Hi-Fi PCR polymerase, which are used to amplify inserts for infusion cloning, is very good. It's very high fidelity. It's also directional, and you can see there are no extra bases being added between the vector and insert, which is great because obviously you don't want to add additional amino acids to your target protein. The other nice thing about infusion is it's very versatile. So it's ideal for several different applications. So apart from regular single fragment cloning, it's also great for cloning into large vectors, such as cosmid vectors. It's also great for multiple fragment cloning, where you want to assemble multiple inserts in a single reaction. It's also great for site-directed mutagenesis, so this includes deletions, insertions, and substitutions. And I'll go into more detail about these two applications in my next few slides. But equally, infusion's been used in many CRISPR-Cas9 cloning experiments, and there are many papers out there that cite infusion for these experiments. It can also be used to add small N or C terminal tags. Really, we believe that with infusion we have such a powerful system that you can design any construct and use infusion to deliver this construct. So first, I just want to talk about the multiple fragment cloning application of infusion, which is a really great application because it allows you to assemble multiple inserts into your linearized vector of choice in a single reaction. And this is not even possible really with ligation-based cloning, just because you're limited by the sequences of the inserts. And often you need to do several rounds of subcloning before getting your final construct. So the first step, as always, is to prepare the linearized vector, either via restriction digestion or via inverse PCR. The second step is to amplify your various different inserts. So here I've shown 3 inserts in this example, shown in red, blue, and green. And here primer design is of course critical because you need to ensure that each insert either has an overlap with one end of the linearized vector or to the insert to which it will be joined. So you can see here that insert 1, shown in red here, has an overlap with this end of the linearized vector, and then it also has an overlap with insert 2, shown in blue here, and equally insert 2 has an overlap to insert 3, shown in green here, and then finally insert 3 has an overlap to the other end of the linearized vector. So of course primer design is very critical here, and again I would recommend you to use our new online primer design tool for this purpose. But the one difference between multiple insert and single insert cloning using infusion is that we recommend using 20 base pair overlaps for multiple fragment cloning. This is because as you clone more and more fragments, the total number of colonies drops, as of course there's increased pressure on the cells. So we found that by increasing the overlap length to 20 base pair for multiple insert, you get a greater total number of colonies, which is why we recommend this. And we also have a specific multiple fragment cloning protocol, so please follow this for the best results. So once you have your inserts and your vector, you simply incubate it with the infusion enzyme. And as before, the infusion enzyme just creates single-stranded regions at the ends of each insert and vector, allowing the inserts to assemble at the exposed 20 base pair overlapping regions. So effectively, 3 inserts are cloned as one insert using infusion. Okay, and this is great because you're not, you don't treat the inserts at all, so you don't digest them, and you're not limited by the sequence. And don't just take our word for it. We have a nice paper for this application. And I just wanted to show you some in-house data for a multiple fragment cloning that we did in-house. So here we try to clone 3 different inserts, MBP, Prof12, and ACGFP, into linearized PUC19, and then used infusion to assemble a final construct of 5.2 kb. So you can see that we obtained 89 colonies in total, and when we screened 26 out of 89 via colony PCR and sequencing, 26 out of 26 were correct. So really 100% cloning efficiency, which is a great result for infusion, and just 1 colony on the negative control, which is good because it shows that there's very little background with infusion. Another nice application of infusion is, of course, that it can be used for mutagenesis. So if we just take the example of substitution mutagenesis, here by combining the power of inverse PCR with infusion, you can easily do site-directed mutagenesis. So if you imagine that you already have a vector that has your insert cloned into it, and you just want to substitute a few bases, which is so this is this region shown in yellow here. All you need to do is first design inverse PCR primers, okay, that sit on this yellow region, and specifically the 5' end of each primer should contain the bases to be mutated, but should also contain a 15 base pair overlap with each other, so that you end up with a linearized inverse PCR product which contains the mutated bases and the overlap. You then simply incubate this with the infusion enzyme, which basically will recircularize the vector at these overlapping regions. So this is a really elegant way of doing site-directed mutagenesis. And similarly, you can also do insertions and deletions using this protocol. And you can read more about this in our Nature Methods app note. Okay, so finally, I just want to talk about the actual product that we provide. So the latest version of our infusion system is called Infusion HD Cloning Plus, which we believe is really the cloning solution to all your cloning experiments. So what comes inside the box is, of course, the Infusion HD enzyme premix, but equally, we also provide a high-fidelity PCR polymerase, our CloneAmp Hi-Fi premix, just because it's very high fidelity, which is important for cloning, and it's also fast. We also have an insert purification kit and our stellar competent cells, which work best with infusion. So the reason we provide all these subcomponents is because they're really optimized subcomponents that work best with infusion to ensure a high out-of-the-box success rate. But equally, outside the box, we provide expert tech support. So if you have any problems with your cloning, you can always contact our tech support. And really, the Plus system is a value pricing, because if you were to buy the individual components separately, you would have to pay much more. And as I said before, don't just take our word for it. We have many, many publications for infusion. In fact, over 1850 papers have cited infusion. Okay, so now that you've cloned your target gene into your expression vector of choice, hopefully using infusion, the next step is to express the target protein. Now, as I mentioned before, we have many different expression systems, but I just wanted to talk in more detail about our B-Satilus system and our HEK293 vectors. So our B-Satilus system really is popular with many researchers who want to do large-scale protein production. This is because B-Satilus is a safe organism, so you can use it for large-scale kind of enzyme production within the food industry, etc. So what comes within our kit is actually this PBES vector, which contains the subtilissin promoter, or this APRE promoter. And it also contains a signal peptide. But apart from the vector, we also provide a library of different signal peptides, so 173 different ones to be exact. So the first step is to clone your target gene into the multiple cloning site. Okay, so you can of course use infusion to be fast. So once you've cloned this, the next step is to digest this vector with MLU1 and ECO521. So this will remove this APRE signal peptide. Okay. You then do a library scale cloning reaction with infusion and this spDNA mixture. So to generate basically a library of your target protein where each plasmid has a different signal peptide. So you'll have your target protein with many different signal peptides, which will allow you to basically select which signal peptide leads to the best secretion of your target protein. And the good thing about B-Satilus is you get high levels of active secreted protein, so you can directly purify it from the medium. Bacillus is also endotoxin free, unlike E. coli. And and you can get a very high yield as well, as I showed in my previous overview slide. So it's really a nice system if you want to do large-scale protein production. And the other system I wanted to talk to you about is our ultra-high expression PHEK293 vectors. So they allow high-level expression of your target protein via the HIV-1-based TAT transcriptional activation. Okay, so as you can see, our PHK ultra expression vector 1 carries this TAT activation element, right, and also the TAT cassette. And of course, you clone your gene of interest into this vector. Again, you can use infusion to be fast. So when you then transfect this vector into your HEK293 cells, which are, of course, mammalian cells, you'll get the mRNA being transcribed. But you also get this TAT activation response element forming like the stem loop structure. And TAT, which is encoded in the mRNA, binds to TAR and activates this. So in this way, you get high-level expression of your protein of interest in 293 suspension cells. And we have a nice online tech note, so you can visit our website to find out more, which use these vectors to express OKT3 monoclonal antibodies. So it's a really nice system if you want to express high levels of your target protein in mammalian cells, specifically 293 cells. We offer two different vectors here. So vector 1 carries the TAR and TAT within the same cassette. Vector 2 provides TAT on a separate cassette, which will allow you to optimize your protein expression, fine-tune it better. So once you've expressed your target protein, often with a tag such as a his tag, the next step is to purify it. And as I said before, most people are using resin-based systems to purify his tag protein. But now I want to introduce you to our CaptureM membrane-based technology for protein purification. So what makes our CaptureM technology different is it's based on a high-capacity nylon membrane with specially modified pores, okay? So we have two different types of CaptureM kits. So one for his tag protein purification, which contains immobilized nickel within the membrane. But equally, we also have kits for antibody purification. The only difference is those kits contain immobilized protein A within the membrane, which binds to the Fc portion of antibodies. But in both cases, you can obtain really fast and easy purification of his tag proteins and untagged antibodies. And in fact, we have three different formats of our CaptureM kits: mini prep, maxi prep, and 96 well plate formats, as you can see in the picture here. So it's as easy as doing a plasmid DNA mini prep, except for his tag protein or antibodies. The best thing about our CaptureM kits is that the protocol is so fast, so the entire mini prep protocol can be completed within 5 minutes. So it's really as easy as this diagram here shows. You equilibrate your column, bind your sample, wash, elute, and you're done. But if you're using our maxi prep or 96 well plate versions, you can complete this within 15 minutes. And our 96 well plates are also suitable for use with a vacuum manifold, so you can automate the step too. And in both cases, the protocols are resin-free and can be performed at room temperature. And the great thing is, because the CaptureM membrane is so thin and the purification is fast, it's compatible with many different additives and lysis buffers. And because of the small bed volume, you get a highly concentrated protein. Okay, so let's just compare how our CaptureM columns work compared to resin-based columns, and what makes CaptureM much faster than resin. So if we take a cross-section of a typical resin-based column shown on the top here, what you can notice is that resin-based columns have a very large bed volume, which of course results in a high capacity due to a large internal surface area. So this results in a high yield of protein. But the downside of resin-based columns is that you get often a slow diffusion of macromolecules through the bed, and this results in long separation times. But we do have such resins. If you're interested in our resin-based products, we do offer our His60 nickel and talon resin-based system for his tag protein purification. However, what makes our Capture-M membrane different compared to, let's say, traditional membrane-based columns, which have been plagued by low capacity and low yield, is that Capture-M contains specially chemically modified membrane pores. Okay, they've been modified to have a larger internal surface area and thus a higher capacity than traditional membrane-based columns. And of course, because of the small bed volume compared to resin-based columns, this leads in a much faster purification due to the rapid flow. But however, I should point out that the yield from Capture-M will never be as high as resin. But the advantage, of course, is that it's much faster and much more convenient. So if yield isn't the major issue for you, then I think Capture-M could be really useful to speed up your protein purification workflow. And this is just a close-up of the Capture-M membrane. Okay, so let me just go through the HisTag protein purification kits first. So the actual protocol itself is very, very simple. So the first step, of course, is to prepare clarified lysate from cells expressing your target HisTagged protein. So these can be bacterial cells, mammalian cells, or if you have a secreted protein, you can also directly load cell culture medium or cell culture supernatant. Okay, so once you have your clarified lysate, you first equilibrate the mini prep column, but the protocol is essentially the same for the 96 well plate or maxi prep columns. The only difference is the incubation time in between the steps. So you equilibrate the column, you load up to 800 microliters of your clarified lysate onto the column, and it's at this step that you really save time because you get immediate binding of your HisTag protein to the Capture-M membrane. This happens within a few milliseconds, whereas often with resin-based systems, you need to optimize this for different target proteins. But because the Capture-M membrane is so thin and flat, you get immediate binding. And if you have dilute samples, you can also do several binds. Okay, you then do a wash and elution step. And as you can see, when we did this experiment with HisTagged GFP and then ran it on an SDS page, you get quite nice clean bands. And just to give you information on the maximum yield that can be obtained with our different HisTag kit formats, you can see you can get up to 100 micrograms of HisTag protein per column if you're using our mini prep columns. And this is the same per well of our 96 well plates, but this goes up to 2.5 mg per column with our maxi prep kits. So, yes. However, for those of you who are doing purification on a larger scale than this and really want much more than 2.5 mg per protein, we've recently launched our Capture-M HisTagged large volume kit. So here, the only difference is we provide our Capture-M kit as a filter unit. Okay, so here the filter unit contains a Capture-M membrane, and you simply filter the sample using a vacuum pump rather than a centrifuge. That's the only difference. So here we used these filter units to actually purify HisTagged Metridia luciferase, which was secreted, I think we expressed it in 293 cells. It was secreted into the cell culture supernatant, and we directly loaded the cell culture supernatant onto this Capture-M filter unit. So you can see we first equilibrated the column with 50 mils of the equilibration buffer. Then we did two bindings of 600 mils of media. We then did a wash step and eluted in 7.5 mils. And you can see that you can get up to 25 mg per unit of protein using these filter units. And equally, you don't need to use cell culture supernatant. You can also start off with clarified bacterial or mammalian cell lysate as input. So this is good if you want a larger scale version of our Capture-M kits. Now, the other great thing about our CaptureM kits is that they're compatible with many different additives. So, for example, EDTA is often used as a protease inhibitor, but really damages resin. So often what researchers need to do is add additives to the lysis buffer and then do a buffer exchange step prior to purification with resins. Whereas this is not necessary with CaptureM because the membrane is so thin and the purification is so fast. So you can see our CaptureM membrane tolerates up to 10 millimolar of EDTA, up to 30 millimolar of beta-mercaptoethanol, up to 10 millimolar of TCEP, and also up to 10 millimolar of DTT with no significant effect in protein yield or purity. And we have tested many more additives, so these are mentioned in this table here. But I also wanted to let you know that CaptureM can also be used to purify difficult proteins such as membrane proteins. So, for example, G protein coupled receptors, which are transmembrane proteins, which are difficult to purify because of three main reasons. Firstly, low expression levels. Secondly, high hydrophobicity. And thirdly, due to low stability in detergents. So how can CaptureM products help with the purifications of such difficult GPCRs? Well, firstly, if your protein is expressed at low levels, often researchers get around this by expressing the protein in a high volume to get sufficient protein. But if you're using resin-based methods for this, you need to use a high volume of resin, and you end up with a very dilute protein at the end, which you need to concentrate. However, with our CaptureM MaxiPrep or large volume kits, you can use them to pass a high volume of lysate several times. So even if you have a dilute sample, you can do several bindings. As long as you don't exceed the maximum binding capacity, this is fine. But then because of the low bed volume of the CaptureM membrane, you will purify a concentrated protein because, of course, you're eluting in a much smaller volume than you would be with resin. Secondly, often GPCRs are not soluble. They have a high hydrophobicity. So often researchers need to use detergents and other additives to make the protein more soluble. This can often be a problem because these additives can degrade resin. So what researchers usually need to do is add the additives to the lysis buffer and then do a buffer exchange before doing the purification with their resin-based systems. But with CaptureM, there's no need for this buffer exchange step because it's so fast and the protein doesn't actually sit on the membrane for very long. And often these GPCRs also have a low stability in detergents, which can be a problem if you're using resin-based methods. But because the CaptureM technology is so fast, the protein time in the detergent solution is minimized to minutes. And in fact, you get an improved reconstitution of the protein after purification and also improved protein functionality. So in this way, you can use CaptureM to purify more difficult membrane proteins such as GPCRs. And this is in fact exactly what we did with our CaptureM his-tagged mini prep. We used it to purify 6 his-tagged adrenoreceptor beta 2, which is a GPCR. So we first expressed 6 hist ADRB2 in 293 cells, prepared clarified lysate from these cells, and then followed the mini prep protocol as usual. We then did an SDA page and blotted onto a membrane. And when we Ponsou stained the membrane, you can see that the 6 his-tagged ADRB2 is present in all the fractions. This is this band here. When we then did a specific a Western blot with a specific antibody, you can see that it's enriched in the iluate. So in this way, we were able to purify this difficult protein in just 5 minutes with CaptureM his. I want to just quickly move on to our antibody purification kits. So the principle is exactly the same, except the membrane has immobilized protein A instead of nickel, and this allows it to bind to the Fc portion of antibodies. And in terms of the protocol, it's also almost exactly the same as the his-tag kit. The only difference is, of course, you load your diluted antibody sample. So this could be serum, A-site fluid, or cell culture supernatant. And again, you get immediate binding of the antibody to the membrane. And you can do several loads if you have dilute samples. but after elution you can see that we obtained quite nice clean bands for the IgG heavy and light chains with very little albumin coming through. We also tested the ability of our capture and protein A mini prep columns to bind or to purify antibodies from various animal sera. So you can see it worked quite well with sheep, goat, rat, mouse, human, rabbit and horse. Although of course the amount of antibody that was purified varied dependent upon the species being used. Other commonly used samples that we tested included mouse IgG1 from hybridomas and human IgG1 from 293 or CHO cells. So here we got a typical yield of 40 to 60 micrograms or 65 to 100, which may seem a little low, but it's pretty good for such samples. And again, this is just the maximum yield that you can get with our different formats. So it's 300 micrograms of antibody per column of the mini prep, or per well of the 96 well plate. And this goes up to 2.5 mg for the maxi prep columns. We don't yet have a large volume version for the protein A kits, but our R&D are working on this. Another nice application of our protein A kits is that it can be used to easily screen for monoclonal antibodies directly from hybridoma supernatans. So in fact, this is what we did to screen for the best monoclonal antibody against Cas9 from hybridoma supernatans. So here we used our capture and protein A maxi prep columns to in fact isolate antibodies from 7 different hybridoma clones. So these are these 7 shown here. And in the 8th lane, you just have a polyclonal antibody. You can see we got pretty good yields. When we then did a Western with these different antibodies isolated from these 7 clones, either against a low, using a lysate expressing Cas9 at a low level, shown in lane 1 here, or at a high level, shown in lane 2 here, you can see that we got different signals from the different clones. So we got the best signal with clone 3 and clone 5, which were then selected for further analysis. We then did a Western using different amounts of recombinant Cas9. So we go from 20 nanograms to 10 to 5 to 2.5 to 1.25. So you can see that clone 3 in the end was more sensitive than clone 5, and then was chosen as the best clone for the most sensitive clone against Cas9. So in this way, we were able to rapidly screen many different hybridoma supernatants in parallel using our capture and protein A maxi prep column. So for those of you who are doing these kind of screening experiments, our capture M kit can really save you time. Another nice application of our capture and protein A mini prep kits is that it can be used for rapid immuno and co-immunoprecipitation experiments. So how is this possible? So here we used our kit to purify or to immunoprecipitate PP2AB subunit. So what we did was we first prepared lysate from cells expressing PP2AB and then incubated it with the antibody against PP2AB. We incubated the antibody with the lysate for 10 minutes with end-to-end rotation on a wheel. This antibody lysate mixture was then loaded onto an equilibrated protein A mini prep column. Okay, so here, not only do you get binding of the antibody, you also get retention of the target protein that the antibody is bound to, so in this case PP2AB. So then after washing an elution and doing a Western blot with a specific antibody, you can see that we were able to pull down PP2AB using this protocol in just 20 minutes. So this is much faster than doing IP the traditional way. And similarly, we were also able to do a fast co-IP. So here we were pulling down known strong interactors P53 and SV40. So again, we prepared lysates from 293 T cells, which we know express SV40T and P53 endogenously. We used the SV40 antibody for the initial immunoprecipitation with the lysate. So after this 10-minute end-to-end rotation, we again did the rest of the binding, wash, and elution steps as normal, and then did an Western blot using a specific SV40 antibody, and you can see that we were able to pull down both SV40 in the elution fractions, but not in the negative control. So here, no antibody was used for the initial IP step. When we then stripped and reprobed this blot with the P53, you can see that we were also able to pull down P53, which we know is a known strong interactive of SV40, in just the elution fractions and not in the negative control, which is good because it shows that you can do both fast IP and co-IP. And in fact, we have a new kit, Capture IP and co-IP kit, which features these protein A mini prep columns, but also includes the buffers required for IP and co-IP. So another new addition to our Capture family is our Capture trypsin kit. So here the membrane contains immobilized trypsin instead of protein A or nickel. But the advantage is that it can really speed up your mass spec workflow, because for all mass spec researchers, you first need to digest your protein into peptides, usually using trypsin, before doing your mass spec analysis. So the conventional way to do this is to, of course, first prepare your sample, then to denature, reduce, or alkylate it to reduce to expose the tryptic cleavage sites. So this can take up to 2 hours, and then to do the actual trypsin digestion using liquid trypsin, which takes typically 4 hours to overnight. But with our Capture trypsin kit, you can completely get rid of these two steps, and you can complete the entire tryptic digest in just 2 minutes. So how does this work? So you first activate the Capture trypsin column by adding 200 microliters of activation buffer. You spin for 1 minute, discard the flow through. You then add up to 80 microgram of your protein sample, so it can be in any standard buffer, onto the membrane. You spin for 1 minute, so digestion is occurring during the spin step here, and you're done. You have your digested peptides coming through in the flow through. You can then carry out your mass spec analysis. You can see with our trypsin kit, you can really speed up your mass spec workflow. And this is just some data. So here we compared our Capture trypsin kit versus in-solution trypsin to digest apomyoglobin. You can see this is the intact protein. We obtained 70% digestion with the in-solution trypsin, so this was carried out overnight as per the manufacturer's instructions. Whereas with our trypsin kit, we obtained 100% digestion with very little intact protein. So you can see it works pretty well. We also tried compared our Capture trypsin kit versus in-solution trypsin for myoglobin, but this time we didn't denature the sample, so we didn't do this step here. And as you can see, this is undigested myoglobin. So you can see when you use the sample as it is with in-solution trypsin, you get no digestion at all. Whereas when you use it, digest it with our Capture trypsin kit, you still get 60% digestion, even if you just use the sample as it is, which is, again, a good result. So just to show you some mass spec data, so the apomyoglobin analysis was actually done by Dr. Bruning from MSU, one of our collaborators. So he ran the peptides generated from the Capture trypsin digest of apomyoglobin on a mass spec machine, and he was able to obtain 100% sequence coverage from these different peptides, which shows that our kit works pretty well. And finally, you can also use our Capture trypsin kit with more complex samples, such as whole cell lysate from BT474 breast carcinoma cell lines. So here, the cell lysate was first was first separated into 19 different fractions via reverse phase HPLC. Each fraction was then digested with trypsin, and the peptides were analyzed by mass spec. And in this way, we were able to identify 2,320 individual proteins, which is a pretty impressive result. And finally, the latest member of our CaptureM family is our CaptureM Exosome Isolation Kit. So we really believe in this CaptureM technology and have invested in it to generate many new products. So for those of you working on exosomes, you'll know that it's quite difficult to isolate exosomes and to get a high yield. So we have our new kit based on our CaptureM technology, and you can use it to get a high yield of exosomes from cell culture supernatants. So how does it work? So this time the CaptureM membrane contains an exosome binding component. So we can't disclose this information because it's proprietary, but it's non-antibody based. But like all the other CaptureM kits, it's a simple rapid protocols. It makes use of maxi prep columns, so you're able to isolate exosomes in under 30 minutes. Without incubations or ultracentrifugation, and you get a very high yield, so over 2 times 10 to the 11 of purified exosomes per column. And it's a complete solution as well, because we also include pre-clearing column to remove large debris, and the actual isolation columns as well. And just to show you some data from the CaptureM Exosome Isolation Kit. So here we did an experiment where we used it to isolate exosomes from cultured HEK293T cell, i.e. the cell culture supernatant. And you can see that after nanoparticle tracking analysis, that most of the exosomes that we isolated had a mode size of 95 nanometers. And a D90 value, i.e. 90% of the exosomes isolated were under 211 nanometers in size, which fits with the expected size of exosomes. And if we compare our CaptureM exosome kit with other competing technologies out there, so competitor T has a precipitating solution for exosomes. And we've also shown ultracentrifugation here, labeled ultra, because this is what most researchers commonly use. And of course, this is very complicated. You need a huge centrifuge. It can take a long time, and it can potentially also damage the exosomes. So you can see, despite using the same input, we got a much higher yield with our CaptureM exosome kit compared to either of these competitors. We see 70% yield versus 10 to 15. But if we look at the size of the exosomes isolated, they're very similar, showing that what our kit isolates are indeed exosomes. Okay, so that brings me to the end of the webinar. If you want to find out more information about any of these products, you can visit our website, so our cloning resource portal or the protein expression and purification learning center. Or you can email our tech support address. Okay, so if you have any questions, I have some time. Just type them into the chat box. Okay, I just have one question. How does infusion compare to Gibson assembly? This is a good question. So the technologies are similar, but the important difference is that you get with Gibson assembly, the enzyme has 5' exonuclease activity, whereas with infusion it's 3' exonuclease. And also with Gibson, you get the gap repair and ligation happening in vitro, which can sometimes lead to more background colonies, both for multiple fragment cloning and single insert cloning. Whereas with infusion, the gap repair and permanent covalent linking happens in vivo in the bacterial cell. So you get less background colonies with infusion compared to Gibson. Okay, I think we don't have any more questions, so thank you for joining us today, and you will receive a copy of the presentation as well as a link to the presentation itself soon. And if you have any more questions, just feel free to email our tech support address, techeu@takarabio.com. Thank you, and wish you a nice day.